Congrats, you've got the perfect co-IP result. This is what a perfect, textbook co-IP result looks like. If your co-IP data turns out like this image, then congrats, the PI in front of the screen is probably smiling already. Let's break down how to read this result. First, look at the IP group. IP anti-GFP means that the antibody coupled to the magnetic or agarose beads is against GFP. The point here is to pull down your target protein. In this case, we're using anti-GFP to pull down protein A. Now, if protein A interacts with protein B, then when protein A gets pulled down, protein B should come along with it. So in the IP Western blot, we should see both protein A and protein B bands in each treatment group. That's your evidence that they interact. But if you only see a band for protein A and nothing for protein B, that means protein A didn't bring B with it, no interaction between them. Now, let's talk about the input group. You'll see different names for this group. WB, IB, WCL, etc., but they're all basically the same thing, a control group. It's there to confirm your protein is actually present in the lysate, positive control, and also to help evaluate how efficient your IP was. Usually, you'll detect your target bands in the input. But sometimes, for example, if your pulled-down protein is modified, like SU amylated, or it's unstable, the band might be weak or missing, so don't panic right away. Then we have the loading control, which is used to show that the protein levels you see aren't just due to loading differences. In this example, after treating the samples with drug X or Y, protein B levels changed, while protein A stayed the same, and that might lead you to some interesting conclusions if you look into it further. Lastly, some labs include an IgG control, super important to check for antibody specificity and rule out non-specific binding. How do you do that? You run another IP using a non-targeting IgG. One that matches the host species and subclass of your real antibody, like rabbit or mouse IgG1, but doesn't bind anything specific. If you see signal in the IgG lane, that means the binding might just be random, like to the Fc region of the antibody or to the beads, not a real protein-protein interaction. That's non-specific background.